Hello guys, how's everyone doing? Can you guys just respond letting me know that you do in fact have audio right now? Can you guys just respond letting me know that you do in fact have audio right now? Can you guys just respond letting me know that you do? Once again guys, can you just let me know that you are watching? That I'm speaking to someone? Can someone just confirm that they hear me? That you're here? That I'm doing this for a reason? Literally anyone, type in anything, confirm your existence. Thank you, Veronica. Okay, thank you, Maliha, Daniela. Alrighty. So, can you guys tell me what exactly you wanted me to discuss with you guys? Any questions that you may have, anything that needs to be clarified? Because I'd like to get started with that, and then we can move forward. Anything that's helpful. I mean, you really only have two major choices, two and a half, you know, because we're going over digestive, we're going over the urinary system, so excretion in general, and how it relates to metabolism, and then you have the small part of the reproductive system. So you guys tell me what you want me to talk about. What is the sphincter called? The one that goes from the esophagus to the stomach, please. So the stomach is called the gastric region because it's going to be secreting gastric juices, and it's going to and thus the sphincter between your gastric region and your esophagus is aptly called the gastroesophageal sphincter. It's also known as the cardiac sphincter because it's going to be the sphincter that is right next to the cardia region of your stomach. So How about now? Hello? Can you guys hear me now? Can you hear me now? Check, 1, 2. Okay, so there's a huge delay. I'm gathering. So, okay, awesome. So you have this cardia region. So I guess I'll start from the beginning. Okay, so you have two sphincters, which are muscular valves essentially, that are going to be, one of them is going to be in the entrance of the stomach between the esophagus and the stomach. And then you have the one that's in between the stomach and the small intestine. So the one that's going into the stomach from the esophagus is going into the cardia, or the heart of the stomach. So we can call this one the cardiac sphincter. Alternatively, this one is called the gastroesophageal sphincter because it's in between the gastric region and the esophagus. Other than that, we have the pyloric zone of the stomach over here, and this is going to be separating, and then we have a sphincter, the pyloric sphincter, that's going to separate the pylorus or the pyloric zone from the small intestine, the duodenum. So you have those two sphincters. Danielle, is it clear now? So as you can see, there's a couple of different names that you can use there. I got you. Okay. Just give me 1 second. I'm looking for a good image. Okay, I think I'll use this one. And that one. Nope, not that one. Let's stick to this, and let's see what this one's all about. Okay, so let's take a look at osmotic pressure. Okay. Just give me 1 second. Make this a little bit better. Okay. So to explain oncotic pressure, we have to explain osmotic pressure for. Oh my God. So because of the humidity, my laptop is just moving the mouse as it wishes and clicking things as it wishes. So sorry for those technical difficulties. But yeah, anyway. So in order to explain oncotic pressure, we have to discuss osmotic pressure first. And osmotic pressure is going to be the pressure created by osmotically active particles. What I mean by that is that you have certain molecules that once they are they encounter water will dissociate. So when they dissociate into ions that are positive and ions that are negative. Because of this, they are going to create electricity because you have positive and negative being generated. Since we're creating electricity, we call these things electrolytes. So things like NaCl, these salts that we have in our sports drinks, we call them electrolytes because they literally conduct electricity. And the way that they do that is by creating positive and negative charges in the same substance. So there is instability or energy that's created there. So these particles, the reason why I'm saying particles and not concentration or anything a little bit more ambiguous is because what matters is the number of particles there. So there's conservation of energy and conservation of mass that's applied to everything in the world. And when something is dissolved into water, there is a conservation of the mass. So like NaCl, 1 mole goes into the water, and then it's gonna dissociate. In total, the number of NAs and the number of Cls and the number of NaCls together, the mass is going to be the same as when you first dropped it into the water. Nothing changed about the mass of the salt in the water. What did change is the number of particles. When it dissociated, it went from being 1 mole of NaCl to being 1 mole of Na approximately and 1 mole of Cl approximately. I say approximately because theoretically they should all split and remain split. But just like Bradylina, there's going to be a little part of them that's always going to recombine if they get close enough. So you're going to have the 10% of those NAs and 10% of those CLs are going to recombine to become NaCl. So you have 1 mole of NaCl making 0.9 moles of Na, 0.9 moles of Cl, and 0.2 moles of your NaCl recombined through random ion pairing. I'm giving you this overview so that now when I talk about why these solutes are going to be osmotically active, you'll be able to understand that. So, now that we know what an electrolyte, we know what dissociation is, we know what an osmotically active particle is, and why I keep using particle and not just concentration or something like that. So, whenever particles that take up a certain space are placed into water, they're going to create an imbalance in the water and salt balance. Your fluid electrolyte balance is going to be disrupted. So water is going to want to follow the salt and go to the place where there's a higher level of salt so that the concentration is going to be the same. So if you look at the top over here at osmosis, the water is moving from a place of it's moving from this place where you have a low number of salt to a place where you have a lot more salt. And the reason that it's doing that is to correct the imbalance across the membrane that exists as a result of the membrane that's separating it. So the salts aren't going to move like you see in the bottom part for diffusion, because that would be active transport. They can't just cross the membrane on their own. They need the presence of that of the water to make it cross over. So the water is going to be the reason why that's going to cross over. I'm sorry guys, my computer's just bugging out. Okay, so in 1 second. Let's see if that fixes it. Okay. So because the salt isn't going to move, you're going to have the water move to fix this imbalance that you see here. So that's what's happening in osmosis to restore equilibrium across this membrane so that the concentration of this section is the same as the concentration of this section in terms of the number of osmotically active particles in a certain volume of fluid. So over here, the solutes move, but the fluid stays the same. In osmosis, the fluid moves because the solutes don't. Okay, so osmotic pressure is going to be higher in places where you have more salts, because that's going to pull water there. So it's a sucking force towards here. This is also the case for your oncotic pressure. So oncotic pressure is describing your colloids. Colloids are proteins. And the main one that we care about is your albumin. Okay. So albumin is the most abundant protein found in your blood. And what albumin is going to do is basically create a lot of osmotic pressure. So it sucks in a lot of fluid. So it's going to make it so that it's a suctioning force for things to be in wherever you have your colloids that create this oncotic pressure. Now, what that means is if the colloids, the proteins, leak out of the capillaries that you have in your glomerulus of your kidneys, you're going to have fluid be sucked in the direction of those albumin. So that's going to be an issue. So what's going to happen is that you have, it would create albumin urea, because the albumin would end up there. And then you would have a decrease in the ability to concentrate your urine as a result of that. Now, all of this is related to the filtration process that we were discussing before, which is going to be the Starling forces in glomeruli. So, at the very basis of this, let's see if we can get you a better picture. Okay. I'm just gonna... Okay. So you have forces that are pushing the fluid out of the capillaries and into the Bowman capsule, which is where your primary urine is being formed. And then you have forces from the capsule that are going to be pushing onto to keep the, pushing against whatever is being filtered into this capsule area. So they're pushing against each other. So that is the hydrostatic pressure coming from your blood towards the Bowman, compared to the hydrostatic pressure coming from the Bowman to the blood. So they're pushing against each other. And then the thing is, though, the hydrostatic pressure, which is the pressure created by something fluid against whatever is enclosing it, is going to be much greater in magnitude for the inside pushing out than from the outside pushing in. So things are going to move out of the blood into the area where you have your urine being formed, because the hydrostatic pressure is greater coming from your inside to your outside. And then you also have your oncotic pressure that is going to be pulling things inside and outside. So at the same time that you have this pushing battle happening between the inside and the outside, on the other side of it, both of them are trying to pull each other through your oncotic pressure. So the summary of both those forces, of the pushing and the pulling, the balance that's created results in filtration. Things are going to move out of the blood into this urine that's being formed. So it's going to be promoting filtration. I know that was a very long, convoluted explanation, and the fact that my computer is deciding to do things on its own is kind of distracting from my ability to explain this clearer. But maybe this will help over here. So you have hydrostatic pushing and you have oncotic pulling. And you have both of them happening from both sides. But the oncotic pulling that's happening on this side is going to be negligible. So most times you won't even see it. Danielle, did I explain that well enough that you understand it now, or do you want me to go through that in a different way? Okay, so let me try and explain this again. So this PBC and this PGC is telling you the hydrostatic pressure. So the more fluid, so think about this. You have fluid filling up in a container. So like, let's say this over here, my coffee is in this container. Now at this point, we have plenty of free space, so the hydrostatic pressure that's being created is minimal. But now let's say that we fill this up to a point that's past capacity. That means that the pressure has now pushed against the walls so much that the cup had two choices. Either it would tear so that or stretch out, or you're going to have it burst out of one of the lids, right? So that's the pressure that's being generated by less and less free space being present. So that's hydrostatic pressure. Do you understand hydrostatic pressure first before I get into this next part of this? Yes? No? Daniela? Okay, so now with the albumin is the most abundant protein found in your blood. And it has a huge molecular weight. It's going to take up a lot of space. Because it takes up a lot of space, it's going to create a bigger imbalance in the fluid electrolyte balance. And osmosis, the movement of water, is to correct imbalances. So the more space that certain things take up, the greater osmotic pressure they create, which is going to suck water towards them. So oncotic pressure is the pressure created by protein colloids like your albumin. So as you can see here, the thing that says pi, that means osmotic pressure. Alright, so I'll just wait for you to respond and let me know if that's still unclear. If not, we're going to keep it moving. I have some questions for the reproductive system. Okay. Yes, no. Are you sure, or are you just saying that? Okay, perfect. Alright, so now let's get to this sex determination process. So, the first part of this whole sexual determination is the parents are going to have sex, and before they had sex, their body had prepared for this because the existence of the species depends on the ability to reproduce. So since that's the case, your body has already been able to start this process of meiosis, where instead of just duplicating your body cells like normally, your somatic cells, you're going to instead be creating these cells that have half your genetic information. And the reason why it's half is because the other half will come from your sexual partner. So during the process, so these lovely man and woman meet, and they start to, you know, go through the whole process of coitus, which is sex, and they end up going through fertilization, where through this process the sperm swims up to the egg, and most of them, a lot of them actually do, not just one. So a few of them make it their way to the egg, and while they're around the egg, the acrosomal reaction happens, which means that you're going to burn a part of the, or enzymatically break down a small portion of the protective layer around your egg cell, and then a special, very strong sperm cell makes its way into your egg cell and combines with the genetic information that you have there, and so you have fertilization. And in that process, a zygote is formed through the union of the sperm cell genetic information and the egg cell genetic information. So now that you have fertilization already happen and the zygote is formed, this is going to determine your genetic, gametic, and chromosomal sex. Meaning that you are going to have a certain karyotype associated with the correct or incorrect chromosomal sex formation. So if this process goes right, meiosis was carried out in both parents correctly, you only get either an XX or an XY. Meaning that your chromosomal sex is either a female for XX or it'll be a male for an XY. So then from there, now that you have this chromosomal sex, now the sex differentiation process starts, because that was only the determination. So, but before I move on to the gene itself, let's talk about these chromosomes. So, if something goes wrong during the meiosis process, you might end up with something called non-disjunction. So while your genetic information is being split up so that you end up with haploid cells, so instead of having your complete 46 chromosomes, you have 23. So some people might end up with 24 or 22. And the reason for this is as they are splitting during meiosis, if something doesn't split correctly, you end up with 2 chromosomes going into one set of your 46 made up of 23 and 23, and the other set will have nothing in that place. So that means that you can end up with XXY. You can end up with XO. You can end up with OY. You can end up with XXX, super female. So all of these non-disjunction related sexual disorders are going to be a result of an issue happening in the chromosomal sex determination because of an error in meiosis. And then all of them will have certain effects on the rest of the process downstream. Now, speaking of downstream, actually, before I move on, is that clear? Yada. Okay. So second. I'm trying to see if there's a good image for other people to be able to see this. Okay, so this is actually perfect. So during a female's meiosis, you have the development of 4 different cells from the original cell that was being duplicated through meiosis 1 and meiosis 2. So during these divisions, you're going to have the development of one primary ovum, and then the rest of them are going to be polar bodies that disintegrate. Now, those words aren't as important as it is to notice the difference between what normally happens and what happened over here, where they did not have a proper disjunction, or basically where they divided, segregated in the correct way. So non-disjunction means a failure to detach. And since they didn't detach, your ovum now has, your egg cell now has double the chromosomes that it normally should have, and your polar body has 0. So this is an example of non-disjunction during meiosis. And this could be a reason why you end up with those other karyotype chromosomal sex determination issues. Okay, so now that we have this happening, on the Y chromosome itself, you have this gene that's called the SRY, which is your sex determining, what's the R stand for again? Oops, 1 second. Let me look that up real quick. I forgot the word. Sex determining region on Y. So this is a specific gene that is going to be found on the Y chromosome. And if it is found there, then you will start to begin the process of differentiation in a correct way, for it to be able to establish male characteristics. And if it's not found, you will go towards the default state, which is female. So in an undifferentiated state, both of the reproductive structures, internally and externally, are going to look the same. You have gonads, and you have these Wolffian ducts and the Mullerian ducts. And based on the differentiation process, you are either going to develop the male reproductive urogenital tract, or you're going to develop the female one. And so the internal and external structures will develop as a result of that and differentiate. So no SRY means that you're going to continue through the female route. So now you are having a gonadal sex that's going to lead to the development of ovaries instead of testes. But let's say you do have the SRY. Now you're on the road to producing testes. Okay, so now you get to the point where it's your external structures that you want to know what's happening there. Are you going to have a clitoris or a penis develop? So part of this has to do with the levels of testosterone or estrogen that are coursing through the body during that time. So if testosterone is being produced from the Leydig cells in your testes, then you are going to start to develop more of these masculine traits, and if not, you are going to have more feminine characteristics. Now, from there, you also have one of the more important steps for external structure development, which is the during puberty, you have this spike of 5-alpha 5-alpha dehydrotestosterone, which is the active form of testosterone that is going to be involved in the maturation of your external sex characteristics. So the development of the penis from the clitoris-like structure is going to be dependent on the fact that you have enough of this 5-alpha dehydrotestosterone. And that's going to be dependent on the ability to have the that's going to lead to DHT. There's the 5-alpha dehydrotestosterone reductase. And that enzyme is what's going to convert your testosterone to DHT, and DHT is what's responsible for external sex characteristics. So in order to have the proper male phenotype as an adult, you're going to have the XY chromosome, which has the SRY gene on the Y chromosome, which is going to produce testosterone from the testes, which is going to be converted to dihydrotestosterone through your 5-alpha reductase, which is going to cause that external sex characteristics to actually develop. And if these sequential steps are not present, then you will have the development of female reproductive structures and female reproductive behaviors and hormone levels. Okay, so is that clear now, Yalda and Maliha, or do you have any other questions about that? All right, so what's next? Sure. All right, so let's look at this one over here. Okay, so you have two different types of nephrons that are going to be found in your kidneys. So each kidney is going to have 1 million or more of these functional units that we call nephrons. Why are they the functional units? Because this is the location of blood filtration and urine production. So this is the unit within your kidneys that allows for this process to happen. And the names of these nephrons have to do with their positioning. So almost all of your nephrons are what we call cortical nephrons. And
So that's 85% of all of your nephrons. And then about 15% of them are going to be found as juxtamedullary nephrons. So there are a couple of differences between the two, both in their position as well as their structure. So, let me tilt that down. Okay, so if you notice over here, we have our cortical nephron that's located in the renal cortex, which is gonna be one of the it's where you have your dark meat. Your dark meat of your avocado. All right. Okay, so this dark meat on the outside, if this was your kidney, that would be your renal cortex. And then over here, we're seeing that your cortical, which is associated with cortex, is going to be where you have your cortical nephrons. And you'll notice that almost the entire loop of your cortical nephron is found in the cortex. In addition, you'll find that the glomeruli of both your cortical nephrons and your juxtamedullary nephrons are both going to be found in the cortex. So if I was to ask you to tell me if I give you a microscope image and I tell you which one of these is the cortex or the medulla, you should be able to tell me that this over here is the cortex based on the fact that you have glomeruli present. And then in the medulla, you aren't going to have that. Okay, other than that, you have the juxtamedullary nephron, which is going to be right next to, juxtaposed to the renal medulla. So the juxtamedullary nephrons have their glomerulus found in the cortex, but then the entire nephron extends into the medulla, and it's found right at the junction between your cortex and your medulla. So another thing to note is the length of the loop of Henle that you see for the juxtamedullary nephron versus your cortical nephron. Cortical nephron has a much shorter loop of Henle as opposed to your juxtamedullary. This is going to allow a lot more of this concentrating of the urine than you would get in your cortical as a result of the longer distance where you can concentrate it. You increase the surface area by increasing the length across which you can have this diffusion happening and osmosis. Okay, so you were asking me what's the difference between the cortex and the medulla. Okay, so your kidney is going to be separated into a couple of different zones. So this image is actually probably one of the best for this. So if you were to take the image of the kidney, you'll notice that you have this outside layer which is your renal capsule. This is the skin of your avocado. This is that dark green to black skin. 1 second. Hunter! Good boy. Alright. So, when you're looking at the kidney, you notice that it has different layers. So, the outermost layer is just this fiber capsule that goes around it. So that's what you're seeing here, that dark brown layer right there. Now, right next to it, just deep to it, is the cortex. So the cortex of your kidney is going to be the dark meat area, based on this analogy. So this is the cortex. And then you have your medulla, which is in the middle, followed by the end part, which is going to be the renal pelvis. The pelvis is where your urine is going to collect, and it's like the pit of your avocado. So once again, the outermost layer, the part that gives it its skin-like brown color, is going to be the renal capsule. Just deep to that is going to be the renal cortex. Deep to that, where you have your pyramids, your renal pyramids that are shaped like pyramids, that's going to be the renal medulla. And then you have the place where urine collects in the renal pelvis, which is just like your own pelvic area. Okay, so Yala, does that help? All right, what's next? You guys have another 20 minutes. I mean, if you want, Ba, 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 ba. Okay. I mean, we just talked about net filtration, but yeah, I'll show it again. Daniela's whole question about oncotic pressure and hydrostatic pressure, that's net filtration. But yeah, we'll get to it. Okay. So before we talk about this whole process of glycogenesis, you need to know what glycogen is, and you need to know what it's being made of in this case. Really? One second. So glycogen is this branched chain of glucose molecules. Okay, well, we could go over it very quickly again, but so glycogen is this branched chain of individual glucose molecules that have been bound together. So this is your tissue box made up of individual glucoses. Glucose, 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 glucose. And it's a way that we package our glucose for whenever we need it, so we don't have a mess in our body where things are, you know, all over the place. Because if I was to take all the tissues and throw it onto the ground, it would be pretty messy. And whenever I do need a tissue, it'd be hard to find one, or I'd be running around in too many tissues always. So we're gonna package all of our tissues or glucose into this nice little polymer. So here is your starch or your amylopectin or amylose, and this is your glycogen, and both of them are polymers of glucose. It's glucose repeated over and over and over again, stuck together. The process of glycogenesis is taking the glucose and then starting to produce glycogen from it by making these bonds in between them. So you have two different types of bonds that will establish the glycogen molecule. So you have these 1,4 glucose bonds, and you have the 1,6 glucose bonds. And those numbers are just referring to the carbon number. So the alpha 1,6 linkages is the reason why you have branching, and the alpha 1,4 linkage is the reason why you have length of the branches. And in the end, you end up with this glycogen. There you go. So glycogenesis is the process of generating, or the genesis of glycogen. And what is it being generated from? From multiple glucose monomers that are being stuck together. Then you said lipogenesis. So lipogenesis is taking what we have. It could be from various different ways that we get to either of these processes, but you are going to produce your lipids from your glucose. So your glucose is going to be broken down into pyruvate through this glycolysis process, which is the breakdown of glycogen. And then you are going to have the formation. So this is going to be the citric acid cycle or the Krebs cycle, aka. And then you're going to have the production of acetyl-CoA, and then that's going to then be thrown into this lipidogenesis process, and eventually, basically, you're going to take the glucose, and through a long step, you're going to end up creating your fatty acids that are going to bind to your triglyceride backbone, and then you end up with fat. I'm trying to look for a good way to show you this. Okay, that's pretty good. All right, let's see if there's an even better one. Nope. Let's see this one. Oh. Okay. All right, essentially, you are going to have a process of producing fatty acid chains, and it's going to be coming in through the result of using your glucose. So indirectly due to glucose. But we have a couple of different ways that we could get there. But this is a pretty good kind of pathway that how you get to your fatty acid. So you go from glucose to pyruvate through the glycolysis process. From pyruvate, you get to acetyl-CoA through the Krebs cycle. And then from there, you're going to use acetyl-CoA to be able to produce citrate, which is going to be, again, converted to acetyl-CoA, which is going to be going through the beta oxidation, which is a process that is going to produce your fatty acids that are going to be bound to your fatty acid chain. So in order to produce, where is it? Okay, so in order to produce the lipids, you're going to need a glycerol backbone and then 3 fatty acid chains. So what I was describing to you before is the production of these fatty acid chains that are going to plug in to your glycerol backbone to produce a triglyceride. And then that is the essence of your lipidogenesis. Both of them are part of metabolism, and there are ways that we're going to store the excess energy that we have for use later. Because as we know, your lipids are very, very good stores of energy, and then you have your glycogen, which is also going to be very good store of energy for whenever you need ATP through the use of glucose. Okay. Okay, so now for your other question, I'll go over it very quickly. I see that you already said that you've figured it out, but let's get into that as well. So excretion, the process of removing your nitrogenous waste through urine, is going to be carried out by the filtration of your blood to remove any of this nitrogenous waste, by converting that ammonia that you have into your urea, which is not going to be as toxic to us as long as we get it out. Okay, so as blood is passing through here, it's going to become filtered out of the capillaries, the blood vessels, and into the Bowman space, which is where you start to collect the filtrate, which is now the beginning of your urine formation. So what we were talking about before with the net filtration rate is describing what is making this process happen. Glomerular filtration. So if we go back to it, we have something called Starling forces. And Starling forces are going to drive filtration. Meaning that you have these two pushing forces and these two pulling forces that both are going to work against each other to be able to create urine by making things leak out of your capillary. So, in addition to your red blood cells, you have albumin, a huge protein that's going through your blood. So whatever leaks out of the capillaries isn't urine yet. It's your primary urine. It's your filtrate. But it still has to go through a lot more of the process before we can call this urine. Aha, finally. Okay, so going through, so we know that our plasma, right? We know that plasma is going to be made up of water. Actually, let me review one other thing then. Okay, albumin in blood. Okay, so your human blood is going to be, this is your whole blood over here, and then it's made of your plasma and your formed elements. So your plasma is 91% water. How do we keep all this water there? Well, we have osmoregulatory proteins and electrolytes. And water is going to follow the salt and follow these proteins because it wants to balance any imbalances that might happen. So, what this means is that within your 91% water plasma, 7% of your blood protein is fibrinogen, which is going to be involved with clotting, globulin, and we have albumin. And albumin is going to make up 50 to 60% of our blood proteins that are found in our plasma. So since this is the case, albumin is going to play a pretty crucial part in how we're able to perform the functions that we have. So if you look at this diagram over here, we have our normal versus our low albumin. The fact that we have this protein here is going to pull the water towards the inside of your blood, keeping it all together so that 91% stays as 91%. The way that it does it is that this is an example of your plasma proteins that are going to, of your colloids that are going to create oncotic pressure, which is going to suck water in. Which is going to be very different than your hydrostatic pressure, which is going to push the fluid out of the vessels. It's trying to break the wall. So the albumin makes the water come back to it. It attracts it back. And then your hydrostatic is trying to push the water out. So because albumin is this huge protein and it's so abundant in your blood and it serves this function of making sure that blood retains all of its water, we're going to make sure that we don't lose that during the filtration process. So you shouldn't have any albumin in your urine. That wouldn't be a good idea. So what that would do is pull in water with the urine. So instead, we have a high oncotic or colloid pressure over here, pulling water towards here. And at the same time, we have filtration happening because you have a push towards the Bowmans that is stronger than the push towards the capillaries. And the pull of the water towards the Bowmans is almost zero, because you have almost nothing that creates osmotic pressure that should be flowing through here compared to what's in your blood. Any questions about that? Or any final questions, period? Because I do have to go now. We'll see, Malia. No problem, Matthew. Have a nice night, man. No problem, Veronica, anytime. No problem, Victoria and Cynthia. Also, how many questions? Maliha and Yalda, I don't know. And honestly, that shouldn't really... Cynthia, I just saw your question. Yes, of course you can see my dog. Hunter, come here. Come here. He was in the middle of a nap. He's reluctantly crawling over. Fine, I'll pick you up, you big baby. This is my son. Dude, I'd love to. I think he would freak out. Maybe, we'll see. He's annoyed. He wants to take his, yeah, he really does. He wants to take his nap. He is 1.5 years old. He was born on January 1st, 2017. I I will try my best to think about how that would work out. But maybe, possibly. Alright guys, sorry, but I am gonna have to go now. I have a wedding to get to. Good night. Good luck, everyone.